Cell-based assays are a key component of the creation of new medicine, from early target and drug discovery efforts through clinical trial and manufacturing support. Complex biochemical processes in the body can often best be investigated and understood in cell-based model systems. So what system will be the most advantaged model for your project? Having developed the cell-based assays instrumental in getting the cancer therapy T-curve to market, as well as the system for rapid analysis of drug combinations that resulted in 3 patents for its expanded indications, I've seen firsthand how critical it is to carefully weigh the advantages and disadvantages of multiple assay systems. The path you choose can directly impact the success of your analysis. Cell-based model systems can include primary cells freshly isolated from patients, cell lines established for long-term culture and experimentation, and genetically modified cells designed for very specific pathways and diseases. Cell-based assay detection platforms can range from cell growth and death to biochemical and immunoassays measuring signaling and secretion. That being said, what considerations should be assessed as part of your cell-based assay system selection? In early phase discovery, a variety of systems and platforms can be used to select and validate targets and optimize the molecules designed to act on these targets. Novel therapies often have limited commercial reagent availability, and much time is spent generating model systems and custom reagents. Cell transfections, Western blot analysis, and proliferation assays are typical procedures for early phase drug discovery. Medium to high throughput automated screens are useful for prosecuting a large number of molecules efficiently and accurately as projects gain momentum. Now, as drug discovery programs enter into clinical trials and manufacturing, the assays require high levels of precision and reproducibility to evaluate activity. Multiple platforms can still be used, but should be selected to ensure the most robust assays possible. Clinical trial support also includes the development of precise and robust cell-based assays for evaluation of anti-drug neutralizing antibodies, vaccines, and drug activity assays from human matrices. Any quantifiable measure of drug activity in a cell-based system may be evaluated for its use as a model assay. These assays are often very specific biochemical measures, such as enzyme activity, protein secretion, receptor phosphorylation, or ligand-stimulated proliferation. Once activity is defined, the assay is validated for its reproducibility in the appropriate clinical trial sample, such as human serum. You must ask yourself, can the measures of accuracy, precision, specificity, selectivity, and stability of my chosen method be performed to ensure long-term reliability of my assay for supporting the clinical trial? With the discovery of new biological molecules as therapy, it has become critical to be able to quantify drug activity across batches and manufacturing processes of the medicines. Manufacturing support requires tightly controlled processes to permit the validation and execution of cell-based analytical assays under GMP. GMP services evaluate drug product, drug substance, and formulations to support process improvement, stability, and product release. Like assays supporting clinical trials, manufacturing support is typically performed in assays with very specific biochemical measures, along with rigorous data analysis procedures to ensure the most quantitative assays possible. As such, parallel line analysis is becoming the norm for ensuring the most rigorous assessment of relative potency between reference and test material. Parallel line analysis confirms that the maximum, minimum, and rate of response of a given batch of drug is consistent and ensures the most reliable assessment of relative potency between batches. This analysis can be applied to the wide range of assays performed in support of product development and release. As you can see, there's a near limitless variety of cell-based assay platforms that can be developed. Each cell-based model system has advantages and disadvantages, and a critical component of proper cell-based assay development is the knowledge and experience to weigh these considerations carefully to select the most appropriate model. BioAgilytix has an experienced team of cell biologists with proven track records developing and validating robust cell-based assays. We can evaluate using absorbance, luminescence, and fluorescence, flow cytometry, imaging, and in any of our diverse immunoassay technologies. To learn more, submit questions or schedule a conversation with me through our Virtual Podium site. And be sure to visit www.bioagilytix.com for more on our full breadth of services. Thank you for your time, and keep an eye out for more of my upcoming Virtual Podium talks.